Oh, it went off. Hold on. Okay, should be good.
Yeah, you could write down 850K. And you could move that over to the USBL.
The what?
You could change everything to
Oh, yeah, sure.
USBL. If I say USBL again, is it clearer?
Roger. Okay. Good?
Yep.
Are you going to keep it off?
Yeah, for now.
Okay.
If we need it, I'll turn it on, but I don't think we need it. What is happening with this? This is wrong. Let's just do that.
Bridge nav, another 20 meters, 145. Might be interesting to explore it that way.
Yeah.
Maybe before we head up.
Yeah.
I don't know if you'll have enough. I'm going to have to
Can
pull
we
the
just give
ship
her without a ship move?
We're still going along the contour here?
I could just give her down.
We still are going along the contour yet.
All right.
Let's see what happens. Roll with the current.
Mm-hmm. Everything's bent over here. Everything's been worked by the current.
Trevor, we had a question for you earlier.
Uh-oh.
What does heavy feel like in the control?
Good question. It feels like I'm not using much vertical thrust to stay down. That's the short answer.
Okay.
So
Thank
it's lifting you up?
Sorry?
What is that?
It's lifting you up? That's like
No,
one of
it's-
those dead sponges, I think.
It is the opposite of that. It is weighing me down. It's full of rocks. All right, that's as far as I can get with no ship move, I think. Maybe
Okay.
a little bit more, but...
All right, let's take a little jog, just a little bit that way.
Sure.
So let me get my right tool and do it properly.
Andrea, can you explain the pebbles in the sandy areas?
114.
Yeah, so a lot of times in the sandy areas, we'll see manganese nodules, which are precipitates of manganese crust over usually a nucleation site, like another piece of rock or a piece of organic material, like a shell or a tooth. And those grow concentrically, so they grow out and they precipitate those. It grows out of the chemical constituents in the ambient water as a precipitate, and it grows concentrically. It grows over long periods of time. It takes millions of years to form them. And they, at this depth, I believe they are rich in cobalt.
Oh, okay. Thank you.
How much cobalt does it take to be rich in cobalt?
I don't know. Enough to make somebody want to build a tractor that goes to the seafloor.
Right.
Wow.
Nice.
That's a really nice leaf sponge.
Yes.
It's really. I wonder what's living in there. I see
Probably-
3 pink.
Probably squat lobster.
And maybe some stars and some shrimps.
Yeah.
Oop, hello, coral.
That one didn't have a lot of precipitate on it.
No.
It looked clean.
Didn't you guys see a field of these similar corals somewhere along the way as well before?
Yeah, we took a Niskin.
Yeah.
Mhm.
Oh, yeah.
From Noad corals.
To all of our viewers out there, keep sending us your comments, your questions. We'd love to know your names and your location. It's fun to know where people are calling us from.
You have to give exact location. maybe like what
My
state
living
you live
room.
in.
I'm in the bathroom at the moment.
Yeah, I had to go to the closet. My mom was yelling at me. It was too loud. I have my favorite slippers on.
Uh-huh. The ones with little doggy faces on them.
Ones with little doggy faces.
Well, I know
There's
there.
a
Oh,
fish.
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 Oh, yeah, fish.
All right,
Or
Aaron, do your
something.
magic.
Is that a Kumba?
He's moving.
Oh,
Ooh,
Cool
nice.
That's a beautiful blackfish.
bonk.
Oh,
Ah,
can we get some
corals.
shots of that?
Ah.
That is really pretty.
It's very hazardous going down slope like this. Okay, come wide.
Can't see what's going on.
That was good following, Trevor.
It's very
There's
scary.
There's another something flowing. Maybe that's just a
Where am I going? I gotta go this way again.
dandelion.
Well, where we are, speaking of where you are, we are, if you're just joining us, we are in the Tamana Seamount in the southern Wentworth chain of seamounts in Papahanaumokuakea, a Marine National Monument and World Heritage Site. This is our 8th dive or 9th dive? Can't remember.
I believe it's a 9. I always get confused too.
I think it's our
I want
8th
to say
dive.
the 8th or 9th. Yeah.
Yep. And we should be, we're coming up to the top of this feature, which is a Goyo, which is a flat top seamount, meaning that it was at the surface at some point, 70, 80 million years ago, perhaps. And that's why it's flat at the top, because it was overgrown by corals or clams, or because the corals haven't been the most dominant organism in the sea for modifying the seafloor over that many million years. We've had clams and other things that have been the dominant reef-building organisms. But so it was at the surface, and it was at likely like an atoll with a lagoon in the center, and then over millions of years it moved away from its place of genesis, its hotspot genesis likely, and sunk beneath the ocean, and now it's a geo, sunken flat top seamount. And we are going to hit the top probably, I don't know, in an hour or so. We're just kind of cruising along a contour here just beneath this this cone at the top. And we're definitely observing some pretty good current up here at the top. You can see that in the corals and the sponges. They're either very robust and attached to the bottom quite well, or the corals are kind of, the primnoid corals look like little candelabra, or look like little menorahs, kind of, with on a single plane. They're pushed over to the side. Everything's pushed over to the side. Look at that. I mean, it's like you get up to the top of a very windy ridge and you see the trees are all bent at a certain angle. That's exactly what appears to be going on down here. And Ken's saying it's a good illustration of the this hydraulic jump that happens at the top of a typographic feature, and the current is accelerated.
Well, that was pretty good. I am refueled.
You're refueled,
Yeah.
all right.
Yep.
And, you know, the cooks were listening in on your talk because you said holy guacamole, and they gave us guacamole. So is that kind of like coding, or like we could just say what we want and it appears? I mean, it seems like magic.
It's a subtle request. You know?
Uh-huh.
Yeah. So
Is that holy Brussels sprouts?
yeah, so we broke 1,000 meters, didn't we? We're 999 right now.
Never to be
Yes.
repaired again.
Uh-huh.
We are coming slowly creeping upwards along
Just
this
getting
contour.
back down below 1,000.
Oh, you had to do it, Trevor, huh?
We're checking to find
So-
a potential ridge edge here.
So at this depth, there's no sunlight, right, Chris?
So Ken was talking about when the last photon of light expired. He had read or heard that it was around 1,000, 1,200. I always thought it was shallower than that. But enough sunlight to support most animals, and certainly enough sunlight to support plants, that's going to extinguish quite a bit shallower, you know. So most algae or corals that rely on photosynthesis are that is their associated algae that's in their cells. They're going to be well above 100. They're probably going to be no deeper than 100 meters. And then there's a couple specialists that have specialized means of acquiring, or getting energy from very low light levels. And then when you get down about 200, 300 meters, you just don't see any type of light dependent animals. There's still light you can see out there.
What is that, Chris? In the
What's
center
that?
where the
Zoom in.
lasers are
That is-
passing by.
Yellow sponge.
What is that? Oh,
Yellow.
is it dead?
Oh.
No, up above.
Oh, that
Oh.
one too.
No, that where the lasers are right now. Is that? Oh, okay.
It is a
dead
dusty demosponge.
demosponge.
What's this gooey thing below, though?
Yeah, what was that?
Yeah, it's,
Oh, these are, what did I hear about these things? They're like brittle star meshes or something else, or worm, or?
is it dying or
No, it's just,
was it bamboozle, but something?
no,
Do fish sleep in those? Like they do those
No.
mucus cocoons in shallower water?
look, no, this isn't the time machine thing where they had those special little things. This
Fish
is,
do sleep in mucus cocoons in shallow water.
like,
Chris, is this a
okay,
time
I get you.
machine?
You're right.
Yeah.
That's right. No, it's not
Don't
a time
trounce
machine.
my dreams
I
here.
trounced your dream, sorry. And we got a little long-legged shrimp next to it. I guess that's where I was thinking, that time machine movie where he went way into the future and they were sleeping in those little,
Oh, yeah.
What's a time
Sleeper.
machine?
yeah.
There's a Woody Allen movie, that one.
It was a different one, but anyway, we were
I-
way off. We are way off
Time
base here.
machine. We did. We went down a rabbit hole.
Rocks and corals and stuff. Here we go.
Yeah, let's get back on topic.
Crinoids. We have some crinoids there attached to that.
Yeah, we could take a look.
Bubblegum
Let's see, it's a bubblegum coral, but
Yeah.
Mm-hmm. Nice colors on these corals in this,
Hey,
this
you bet.
dive.
Whoops. Okay, you can zoom in there, please. Who's driving this thing? Wow.
see
this
their
Nice contrast.
so you see their feet, and you see how their feet do wraps around the branches.
Mm-hmm.
That's called prehensile cirri. They're not real feet. They're called cirri, and some of these feather stars have prehensile ones where they can make multiple wraps and others don't. And that's actually a helpful little hint as to what the crinoid is. Anyway,
Sure.
We have a question from Australia. How
Okay.
does life like sea sponges grow on the rocks?
well, first they got to attach so that they don't get blown off the rocks, and they attach very early. Their larval stages manage to get down and attach, and then they just start dividing, and they start very slowly feeding until they just get bigger and bigger, I guess.
Thank you.
So the coral that populated, I think the question is, you look at these fans and you're going, well, most of the animal's up and it's on the stalk, but it started out as just a tiny little larvae, and over quite a period of time, then it started secreting some of its hard skeleton and dividing and producing more branches and more cells till it finally looks like this fan shape.
Uh-huh.
Yeah, and I think you explained it really well the other day. These larvae have some ability to do kind of chemosynthetic analysis, or to look at where they're going to settle out, whether it looks good to them or chemically it seems right, and then they can settle, and if the current isn't too strong, then they can attach and then grow up and divide and become larger organisms.
Yeah. It's a mystery. When you get a whole little grouping of the same type of coral like we've had here, this Calyptrophora, Why? Are larvae detecting, via chemotaxis that there are other members of its species around and they settle there? Or is it just one big settlement event where you have a pulse of larvae that all come in at the same time?
And do they stay at that level if they release larvae and they're drifting in the column? Do they stay at this depth, or do they go up to drift in the plankton and then settle down? Or
no idea. It's just larvae and egg and dispersal of all that is impossible to measure. So people try to model it, but they're models.
Oh.
It's not based on real observations.
What's up? Yeah, dew bubble. Yep.
A tiny purple thing.
Tiny purple thing where?
To the right of your green lasers.
Really green. This guy?
Yeah.
Wow, how shaky can I get this thing? Let's see.
The sponge.
Zoom in, please.
Yeah, I actually saw it.
Nice.
There's something else I'd like to take a look at too.
Cool.
Anticlimactic.
Okay. Come wide, please.
Tiny sponge.
Okay, and then to the right there was something right on off screen. Is that just another crinoid, or what is that? Yeah, maybe we could take a look at this. I think it's a different one.
Okay, zoom in there, please. Bonk. Look at that.
It's moving.
That's what I'm looking at. See the very long tips of their arms that don't have any, that they're naked. They're tiny little threads.
Right.
So
that's vindictive.
And the cilia are pretty long. You can see them right underneath the arm.
Yeah, looks like it's walking. Almost.
So I think this is a Telecrinus because those are characteristics of that group.
Are we sampling it?
No, thank you. But it's just a cool observation. We haven't seen one of these so far.
Cool.
Thank you. I'm going to check my thing here to, okay, that should be on the top. Well, now it's called Paratelecrinus and Paratelecrinus conifer.
Got it.
Okay.
Thank you.
There you are, this guy, for you to take a look at.
Yeah, that's really beautiful.
Bubblegum coral.
Paragorgia.
What is that? Right. Oh, that's another one of those crinoids, isn't it, that we collected?
Yeah, I think so.
Yeah, okay.
Looks like two stalks. Am I seeing that right?
I don't think so. Dan got some really marvelous close-ups of that guy.
Okay, zoom in, please.
I was explaining to him when he was seeing the other side that it's like a hybrid between a stalk crinoid and an unstalked crinoid. It's like half a stalk.
What are those two dangly bits in the back there?
Those are, trying to remember if they're also called cirri. I can't remember, but we suspect he can walk around.
Oh, that's his feet.
Yeah, it's his feet, and he's propping, but he's got a stem that kind of comes down and lays flat against the ground.
Oh, okay.
It's a really weird guy, so it's a good collection.
We have a question from Miranda in Portland.
Okay.
Is there a chance that feather stars evolve from organisms like that crinoid with its stem, but just adapted away from being anchored to the strata?
Great question, and that would be my guess. You know, I don't know, of course, but that's a very reasonable guess, I think.
Yeah, thank you, Miranda. And then a 13-year-old wants to know if we have found any new species of fish or coral.
We haven't found any new species of fish because they're really hard to collect.
Yeah.
And corals, yes, we have.
So, yeah, sure.
What, is that a fish? Or, no, that's something purple.
Purple. I'll look at the purple thing.
I think it's the same thing.
Well, let's take a look at the the purple, because I didn't get a really good, oh, I
You're
see,
clear for a reset.
in the background, right?
Yeah, in
Oh,
the
the
background.
lasers.
Yeah, that's Victor Gorgia, but it's good to get Trevor to reinforce that name in his head.
Can you zoom on the Victor Gorgia, please?
Yeah, way to go, way to go.
I mean, you just said it, so.
That's okay, but that's all right. That's,
This one is really deep purple.
yeah,
Deep
I
purple.
think the first one that we saw was almost lavender.
yeah.
Star on the coral.
God. How do you even know that music? You're way too young.
Timeless.
It is.
No, I am.
Oh, you are? Okay, sorry. Let's see what else here. It's kind of like a treasure hunt. There's not tons of things, but then they're all around here. You gotta spot them.
Tiny little things.
Yeah, they're spread out.
Nooks.
So the viewers are saying that there are these little pentagons on the ground. What are they?
Pentagons where?
Pentagons. I guess the white spots. Pentagon shapes. I was thinking it was like the leftover stock of some sponges.
Here's a little urchin. Let's zoom in on the urchin.
Yeah, let's zoom on the urchin. But actually, there are little sea stars that are like that too.
Maybe.
Yeah, so this thing right here, this urchin is called a spitodiadema, and you can see how long the spines are on this thing. It's got a little tiny spherical test and then super long spines.
Thank you.
Yeah, thank you. There are some star-like things that are the stumps or remains of corals and sponges that had just remained on the rock, but there are also both brittle stars and sea stars that are hexagonal as well, and so sometimes we get fooled, and I was fooled last time. I thought we were going to zoom on a sea star, and it was just a smudge. There's a crab down there. I wonder if you could zoom that crab.
I'll stop. Ah!
Let's, oops, if you can't, they can't, but...
Oh, of course. Just there's some serious momentum. He's hiding now. Come out, wherever you are. Oops. Uh-oh. He's fine.
These are just kind of cool,
zoom in, please.
Cool crabs. There.
Nice.
So
right, thank you.
it's a munidid, I think. Munidity in the family Munidity, probably Munida, but that's about as far as I can go. They're Galatheid squat lobsters.
So what are they eating down here?
They pick stuff. I don't know. They catch things. I don't know what they eat.
There's another one in that Primnoidae up there. Are those brittle stars? It's hard to tell.
They're basket stars.
Basket stars.
Yeah, they each have one. If you notice, there's quite a tight relationship.
A viewer wants to know if the corals here spawn like they do in the tropics.
They spawn. We don't know much about it because it's really hard to study coral spawning at this depth. But are they all timed? So you have a big, huge pulse of eggs and sperm in the water, like you do in shallow water in the tropics? We don't know. Possibly, probably, I would think. And some of these corals, they have internal fertilization. And what they release into water is actually competent larvae. They're not releasing eggs and sperm into the water. Fertilization occurs in or on the actual coral. And then they release these larvae. And the larvae, if they want to, they can just go like, you know, a meter or so away from their parent and settle down and start growing up. And we suspect that's the way that you get very dense communities, actually, that you have a lot of settlement events right next to your parents. That's a leaf sponge, Tretopleura, and amongst some demo sponges, those other white ones. That's a Paragorgia, that white Paragorgia to the right of the Tretopleura that we already imaged earlier.
The white Paragorgia is very less appealing than the pink one to me.
It's not quite as, well, if you like purple, you're going to like ruby red too.
Just like the colors, you know?
Yeah.
Oh, bonk.
Oh,
I haven't seen
look
one
at
of those
that.
for a while.
Sure, go ahead.
Go ahead, zoom in, please. fish up there.
This is Iridogorgia magnispiralis, and he's got a shrimp on
Oops,
there.
sorry, come wide.
Okay. There's your pretty little Paragorgia. Didn't see the fish. Was that one of those?
I didn't see it either.
Oh.
We're moving fast. Wow, look at that current. Jeez.
Yeah. There's another Iridogorgia-like thing. A little
Look
bit
at this
different.
coral sponge action up here. Is the coral growing on the sponge? That would be
No, it's behind it.
bonkers.
It's behind it.
You sure?
Oh, yeah, and there's a sea star over to the left there.
Sea star to the left. Let's zoom in on the sea star to the left.
Yeah, I'm sure. But you want to put some, say, a box of Smarties on it?
Yeah. On what?
On whether that the coral's on the sponge or not.
No, gone too far.
Oh, okay.
Oh,
All right. I know this guy. I got to look him up.
It's a cool sea star.
Right, Antonella, I'm gonna back away.
It's a Goniaster. It's in the family Goniasteridae.
Oop, oh, I'm gonna shake a bunch. It's already a little more greeny gray. Now it's, well, see, it's losing its color there. All right, come wide, please. You see that?
Oh, I know this.
Hmm?
Oh,
I know this star.
is it the Bathysera?
Yeah, I got lost its reds.
Bathysera Bathysera?
You know what I mean?
Where are you?
Is that
Oh, yeah, you got it. Oh, I blew right by that one. Way to go.
Goniasteridae Bathysera.
Yeah. So, show that too. I'm pretty sure that's what it was.
Yeah, it
Yes.
looked very,
It looked very
Similar,
similar,
yeah.
yeah.
even with the curled up little arms.
A lot of these Calyptrophora.
so for our viewers at home, you following us on nautiluslive.org, on the main page there is live data showing how deep our ROVs are, along with the temperatures of the water, and also the latest observations. You can see the names of the organisms we are viewing there. So thank you for following us at nautiluslive.org.
Oh, ooh, another shark.
Another shark. Same thing, the Centrocilium, I'm pretty sure.
That one looks a little beat up.
Yeah. Oh, I see on the IPAC, Aaron's got the edge of that cone that we're about to go on.
So do you want to, after this last like contour parallel move, do you want to start chugging up towards your goal?
Yeah, yeah, just to see what it is. That would be great. Thank you.
Yep.
Doesn't look like much based on the contour lines, but oh.
It's going to be very subtle. It's less, there would be another contour if it was 10 meters different.
Yeah.
So there's subtle topography change there, but not, I don't think too much.
Okay, thank you.
It's starting to flatten out a little bit. This is really interesting terrain. You know, just
Yeah.
like,
I don't get it, actually.
no, I mean,
know.
there's no carbonate cap here.
Right.
Nothing yet, that's for sure.
Could we zoom the whip by any
Bamboo
chance?
coral.
Yeah.
Yeah, just to confirm it's bamboo. Since we haven't seen it on the top, I don't think, or maybe we did. I don't think so, though.
Okay, zoom in, please.
And yes,
Bring
it
it
is a
closer.
bamboo. Yeah, there's a node right there.
Whoop,
Mm.
whoop.
Okay.
Okay, stay zoomed in a sec. I'm going to back away. Okay, come wide, please.
So now we can go to bamboo.
Mm-hmm.
I don't know those guys. They're really complicated.
All right.
How about, sorry, the sponge right ahead? I'm sorry.
Sponge
It's
on the
okay.
head.
Yeah, this guy
They
right
were
upside
going to
down.
go too.
Upside down boot.
We're going to start moving 205 towards that goal.
205.
You want to keep the steps smallish?
Yeah, sure, or whatever science wants.
Okay.
Trevor, I got it now. I thought this was a base sponge, but it's not. We've seen this before.
Okay,
So whatever direction you need to go.
we're going this way.
Alrighty, thank you.
Bridge nav, can we get a 20 meter step bearing 205?
Do I have Doppler? I have Doppler. I can do this, right? I'm going to ditch the plate.
There's a tiny little coral right next to it. I don't know what that is. Is that a little corallium or something? Coralliid?
Trevor's just getting ready to
Oh,
drop
okay.
a plate and then check it out.
Okay.
So, Chris, do all deep coral require a strong current to survive? That's a question from George in Florida.
I'm not sure all of them do, but a lot of them do, I think. But there seems to be a number of species that can survive with relatively low current
So he
flow.
was interested in that coral to the left of the sponge there.
Coral to the
Little-
left of the sponge.
Yeah, maybe the ones that eat the smallest organic particles or something. I'm not really sure.
Thank you.
Yeah, it's a good question.
Oh, that's much better.
Oh, and there's a, well, we can look at it as you pull back, but
Go ahead and
there's
zoom in there, please.
interesting chrysogorgid. Yep.
Corellia?
That's good enough, yeah. Coralliid.
Corellia is what I meant. Yep.
And then to the left, there was one of these that's behind you a little bit. It's coming up. Wait for it.
Waiting.
Wait for it.
This?
There it is.
It's hard to see amongst the rocks. Like
I
with all
know.
the-
These are so wispy, and
You can zoom in a bit more, I think.
it's some type of chrysogorgia, I think, but I can't go any further than that.
Looks like a crinoid in the back there. Or,
Yeah, let's, well, you mean it's on it?
yeah, I think so.
You could zoom. I can't see what it
Zoom
is in
more.
the back.
It is.
Yes, it is.
Oh,
Wow,
spot
good
on.
eyes.
Oh, good eyes. Yeah.
All right, come on.
Oh, whoa, wait,
Oh,
wait.
don't come wide.
Look
Stay
straight
right
down.
there.
Stay down at the top,
Pentagons.
straight. The Pentagon.
Oh,
Oh,
those
they
little
are
pentagon,
tiny.
Yeah.
white pentagon.
Ah, who's driving?
There he is, right there.
They're so small.
Oh, there's
What are
several of
those?
them. There's three, at
One,
least
two,
three of
three,
them.
four.
These are really cool. These are brittle stars of all things. They're so weird.
Give more zoom.
Yeah, there is this viewer that keeps insisting about these pentagons.
Yeah.
Yeah,
There's one
so
now.
Yeah.
There's another one.
Yeah,
who's-
and in 2018 we collected
oh
one of these.
wow.
The one on the left is on the go.
Good
Oh
job.
my gosh. Such a
Yeah.
big world
Who's
for
that
such a small little
viewer?
star.
Write your
Yeah.
name. We want to know who you are.
So that brittle star is called Astrophiura.
All right, come wide, please.
And Chris Ma, the Sea Star guy, was really interested in these things because they're quite a fascinating. Wow, that zoom was a twofer.
Yeah, that was.
Well, it was a threefer, I think, wasn't it? Doesn't-
Astrophiura.
Astrophiura. Pretty cool name, don't you think?
Yeah.
I think I'm just speculating here, but I bet the Astrophiura means it's a sea star like Ophiurid or a brittle star. Maybe. I don't know.
A viewer wants to know what was that plate dropped for?
There's too many rocks in the ROV right now, but I mean, I can let Trevor answer that if he wants to.
Oh,
I was
yeah.
thinking we were going to drop the plate to have a little dinner party, but, you know, have other people included too.
By the way, how about the sponge? Could we get that?
Yeah.
It's the first type of sponge like this that we've seen.
Yeah. Go ahead and zoom, please.
That's very cool.
Wow, look at all those tiny little holes.
Yeah, and yeah, nice and tight if you can.
Yeah, go ahead all the way if you got it.
Oh, wow. Is it sealed up on top? Is the?
Ooh.
Yeah, that's called the sieve plate over the main osculum.
Oh,
right there.
wow.
That's pretty great.
And there's creatures in there
I was gonna
too.
say that
Yeah.
cage keeps them in. Oh, it's one of those white shrimps.
Inside.
Yeah, that's correct.
Feed me.
This is the Hawaiian version of the wedding sponge with the two
Wow.
shrimp that are for life inside the sponge.
Oh, what is it
Wow.
called?
It's, well, the genus of this is Regadrella.
Crazy. I'm going to go down to the bottom. I'm getting a little far away.
But that's very cool.
Mm.
I don't know why I was trying to look up what that sponge was the other day. The wedding Venus. What do they call it? Wedding Venus
All right, come
something
wide,
or other
please.
sponge.
Two 205.
Mm-hmm.
No, we're just gonna go
Oh.
with this as the name. This one doesn't show this example doesn't show the shrimp in there.
Regadrella.
Yeah, but it could,
Regadrella.
it's, I think so. Let me check something here real quick. Let's see. Yeah, gonna have to go with that. Get that picture back to show Kainalu. Where was that? Maybe
Thank you, Chris.
Okay, so Connor kept pointing to those little brittle stars, Astrophiuria. And he says that they've been in the background like 80% of all the zooms. It was driving him crazy. He wanted to know what they were. Thank you, Connor.
Yeah, thank you. Really appreciate it. The one time I thought I saw one of those, it was a bust. It was a sand pocket. I thought my eyes were deceiving me, but he was absolutely right.
Yeah,
I'll bet they're all over the place.
this is one of the things I love about our viewers, because that's another extra set of eyes to contribute to our exploration in the deep sea.
No, it's sediment. I'm going to get fooled again. Some of these little sediment pockets can look very symmetrical and geometric. So we're kind of on the edge of some weird thing that looks a little bit like a cone. It's not very well defined.
Tiny star.
One of those little white Pluricralium porcelainum.
You say porcelaina?
Yeah, porcelainum.
Is that because it looks like porcelain?
Yeah, it used to be something that's called Kishinoye, presumably to honor Dr. Kishinoye, and then it was changed fairly recently.
I like porcelain.
We're getting into it looks like a manganese nodule
Oh,
field.
yeah. Isn't that kind of unique at this depth?
It's not so unique on the tops of these geodes because they're flat and they're often sedimented and they provide the conditions conducive for them to form.
Aaron in video, I think this question is for you. Are you able to see clearer feeds in the van than transmits live, or does everyone on board have eagle eyes?
We each have different types of screens and stuff in the control van, and I have pretty good ones that I can get my face about an inch away from if I'm trying to focus on something quite small or something like those corals that are actually really hard to focus on. The ROV pilots have huge screens in front of them that are more for their jobs. So it just depends which screen you're at.
Oh, look at all
Thank
those
you.
stalk crinoids.
But we do have a better view than people who are watching it live at home, just because our feed's coming directly up and not to a satellite and back.
Yeah, and then your eyes are more trained.
Right here. What is that?
That's an old base, I would think, but let's find out.
Yeah.
I'll bet you Smarty's on it.
No. Wait.
Go ahead,
Let's zoom in and then I'll bet you.
Yeah, good thinking.
Nah, I don't think I'll bet you.
Is that an old stock?
I guess so.
Looks like it.
Sure, let's go with that.
Could we ever like scrape what's on the top of this rock and see
Yeah, you
and
can-
bring it for microbiology analysis?
that would be pretty tough, I think. That would be hard to get a specimen like. I mean, the only way you could do it is scrape and slurp simultaneously, I guess. I don't know.
Okay.
Pretty cool little nodules. We're going inside this kind of small volcanic cone, very low relief feature, and there's nodules in here, which is pretty interesting.
Yeah, it's like nodule heaven.
Can't get no relief. Here's a stock sponge for you.
Well, then we can't pass it up. Okay,
Is
what
that
are
Zoom in
you?
there, please.
Bolosoma?
Nope. Steve Reich 1. Andy, do you want to take a stab at this?
Stock
Oh.
Euplectellidae.
Okay, you're there. You're in the right group.
And it is
Sacrocalyx?
yes.
You got it. You
Oh
beat
my
him. You scooped him.
Well
Sacrocalyx.
done.
Well done.
Nice.
White sponge.
You're
So-
beating out
That
the
was
science.
a very good guess.
So we're calling all of those Sacrocalyx. It's a little
Calyx.
yeah, Sacrocalyx. So it's kind of shaky, the IDs, but they
The
all
calyx is the stem or the...
No,
No?
it's I'm going to see if I can find it here, picture of it for you.
Pedunculatus.
Yeah, Andy, you've got a picture of it, obviously. So that's the guy. I think just Sacrocalyx spa would be good enough for that one.
How would you describe that shape?
So, well, it's got these tube protrusions out of the side walls on it, and that's to increase surface area, I assume. And then it also has a big osculum on top, and it's on a stalk. I don't know how you describe that. A really bumpy.
Yeah.
About how many species of sponges do you think we've seen in this
Oh, what was that to the right?
cruise?
You see it?
A fair number of them.
To the right, I think was another one of these ones, these
Oh,
crinoids.
it's still crinoids.
Yeah. That's another one.
Something to the right there. Spongy.
The little guy.
Yeah, let's take a look at that. That's
Take a look at the little guy.
maybe different. And the one in the back afterward with the white
Okay, zoom in,
star or
please.
something.
Oh.
Oh,
that's
could we?
beautiful.
Sample?
Oh.
Well, I know what it is already. So this is Lefroyella. And the reason I know what it is you see all those little orange things in it?
Oh,
Those are commensal anemones
Oh, neat.
that actually live in the tissue, and they can form little
Okay,
tubes
come
on
wide,
it.
please. You want to check this one out too?
Yes, please.
Okay.
Oh, we have a Lefroyella. That's the first one we've seen as well.
This is
It's
interesting.
very thin and
Yeah.
fragile.
Sea
looking.
star all the way up.
Another
Whoa.
Go ahead, zoom in,
Walteria.
please.
Oh, that's a really
Oh,
nice
that's
picture.
a different one. It's got the filaments in it.
Yes. And that's Henricia, the sea star on it.
I have more zoom on this too if you want it.
Oh,
Yeah,
yeah.
if you could zoom a
Go
little
ahead.
bit further on the sponge and sea star. There we go. So this is the difference between some of the Walteria that we've been seeing so far. This is Walteria flemingi, and it doesn't have round holes. Instead, you just see the framework itself, where these are different spicules that are all sort of glued in a mesh on it.
Mm.
And then you've got all these fibers lateralia coming out of the sides of it, but you don't have any more silica applied to the frame, whereas in the other species of Walteria that we've seen this whole cruise, they have round pores because they're adding silica to these openings.
Mm.
So this is actually the type of sponge that the genus Walteria was named for.
Thank
it was
you.
named. Yeah, thank you. That was great.
And bamboo coral.
Yeah. Well, this isn't going to look like it because they come in a lot of forms, but that's the name.
Thank you,
Okay,
Chris.
and then, you know, Walteria and just its flemingi is the second sponge that we saw. Just add that to it. Boy, we are seeing a lot of different things.
Mm-hmm.
Hmm. Are we almost at the top, Chris?
What's that?
Are we almost at the top of the mound?
I think we're actually going down inside. What do you think, Aaron? Are we, I don't know what we're...
I mean, yeah, we're at the top, but there's going to be these little variations in topography, right? As
I
we-
see.
As we go, I mean, we're within the, within the shallowest contour. Actually, it might be a little shallower all the way down there. Top in general, maybe not the tippy top.
Mm.
Not tippy.
Not
just
tippy, just top.
Well, I'm sort of wondering whether or not we ought to just run along the rim, or should we take a look inside? I'm a little torn. It may look just the same, may not be any different, but if Yeah, if we run right where your cursor was, a little bit over to the east, and just run along those contours there that you got there. Would you interpret that as sort of the rim, Aaron, I guess? Oops, I can't hear you, sorry.
Sorry,
Yeah.
I was talking to myself.
Yeah.
It does look like a subtle kind of rim feature, I guess, along there. That's what you're talking about, right?
Yeah, I guess so. Maybe we'll just target that and just run for it a little while and see if it looks good.
We got time. We could
Do you want any kind of sediment core? Or
Any kind of sediment core.
any kind? Like we got many choices. We got the 1-inch diameter core. We got the 3-inch.
So Beth is going to have, it's 1,000 meters. So our recovery time is going to be a little better than normal, right?
Yes.
Maybe an hour?
Yeah, about an hour.
So that gives her 3 hours, right?
That's right.
I'm going to let her decide if she wants to give it a go. Thank you.
Steve said we're still looking to recover at midnight.
Yes. Yeah, okay, that's it. Thanks, Steve. Oops, and there's Asako. Boy, I've been ignoring the... Oh, Timothy Swain said the wedding...
Fish. Sorry.
Yeah,
Left
okay.
corner.
Oh.
Yeah, let's take a look at this.
Let's take a look.
Ooh,
This guy's different.
that is different.
It's going to be a rat tail.
Okay, zoom in, please.
Oh, nice.
Ken, are you out there still?
Look at the size of that eye. It's incredible.
You can really see that eye, Trevor.
He
He doesn't move his eyes, though. You see that?
Yeah.
He looked left and right. Kind of cool.
I think this is another Coryphaenoides, but
He's picking up speed.
he's got pretty long...
Oh.
Oh, bonk.
So Timothy Swain
was good,
actually
Trevor.
identified that wedding sponge, Euplectella
Was
aspergillum.
it?
And I actually checked to see if we had a vase-shaped Euplectellid in our
Where are we
guide, and we
going?
don't, because I guess that doesn't occur here.
All right,
And
some turns there.
Asako Matsumoto is asking, I apologize, I haven't been checking the chat very often. Asako, the dive will be extended until midnight here, Hawaii, which is going to be in about, what is it, about 6 hours maybe? About 6 more hours here. I'm wondering about that rat tail. Could have been something else too, but I'm going to go with Coryphaenoides. And oh, Ken's typing, so great. Maybe he'll have some insight on that guy.
Fish ahead.
Where's the fish? I don't
It's
see
up
the fish.
near the lasers.
Oh,
Oh my
the
gosh.
wee gaffer.
Where?
Are your eyes touching the screen? Oh, it's a shrimp.
And
Wow.
no, apparently not.
Oh, it's drunken shrimp. Canthophyra.
We haven't had a shrimp soon lately. Let's do a shrimp soon.
Yeah, let's do a shrimp soon.
Oh, there's two. Let's.
No, let's do the drunken shrimp. He's prettier. And
Drunken shrimp. What's the scientific name for the shrunken shrimp?
yeah, it's a Canthophyra is the name. I'll find them in the guide so you
I
can
think
spell
I
it.
saw it.
Oh, I bonked. Sorry.
Still good.
There you go.
Oh, thank you.
And that's him.
Thanks, Chris.
Yep. Now, what new critter will suddenly appear?
6-gill.
It could. We absolutely could.
We're in the 6-gill zone.
Yeah. I think this is a little deep for something called a cook shark, so we may not see those.
Mm.
But 6 gills, it could. We could have one wander through. How about that little sponge on the boulder? That little transparent guy right there.
Do you see that?
Yeah, Roger.
I think it's a, dare I say, a polyopogon?
Dare you say? Okay, zoom in, please.
It is not. Could we get a close-up? Well, it might be, but let's see. What are you?
Bonk. Rats.
Whatever it is, it's not doing too good.
Not necessarily, because the roselids can have a very dirty look to them like this. Could we circle around that by any chance?
Yep,
This is
come
pretty
wide, please.
weird.
Full wide on the zoom.
Oh, yep.
This is an oddball. I
Okay,
think
zoom
it,
in past the porch, please.
Kent
There,
said
yeah.
maybe Coryphaenoides, but, or it could be a Kumba. Yeah, he's a little odd to me, Ken. So I'm not 100% sure it was Coryphaenoides, that rat tail, but now we're looking at the sponge. It's the what the sponge?
Mm.
What do you think?
It's pretty unusual. How about one more zoom? And
Sure, go ahead.
I think I'm going to request a collection here, if
Collection
that's possible.
request. Ah. Okay, well, we can set up for a sample.
Yeah,
Come full wide, please.
Yep.
yeah, that's weird. That's weird for even for a nematid or a polyopogon if it's in that group, and it's weird for a roselid. So it's just plain weird.
All right, we like weird.
See some dead skeletons scattered about.
Yeah, dead leaf sponge. And,
Must be fairly
the right.
recent. They're not manganese crusted like some of the others we've seen.
yeah.
All right, that looks pretty stable. I get it, yeah. Okay, starboard box for this one. Bravo or Charlie, I believe.
Yeah, either one. We got Bravo and Charlie open.
Okay. Let's do one more real good zoom on that, please. Go ahead.
I don't know what this is.
I'm gonna do a camera bump down.
I know it's Lophophytus. It's attached by those threads that grapples the substrate. You can see that.
Cool.
And that's it.
Okay, come wide, please, or semi-wide.
Yep.
Good there.
Mm-hmm.
It's a glass sponge.
Why can't you just take the whole
Looks
rock?
like that.
Also, before we leave, if it's possible to image that brown corally thing on the left too, that would be great.
Yep. Remind me.
Okay. Very small. He's going to be squishy, which you can tell already.